Bismillahirrahmanirrahim. So in this video we are going to do with the cell surface contacts or the cell junctions. There can be two types of cell junctions. One are general ones and there are specialized one. In the general adhesive junctions there is no specialized structural characteristic, but in the cell specialized junction they are different type, they have special features and they can be differentiated from each other. The general ones, they are also known as the cell adhesion molecules, and they have three domains. One is an intracellular, one is a transmembranal, and one is the extracellular. Let me show you. So look at this diagram. Consider this the exterior of the cell, this the interior of the cell, this is the cell membrane. So this is a general adhesive junction, which is also known as a cell adhesion molecule or CAM. It has an, on the interior side it has an intracellular component, on in the membrane it has a transmembranal component, on the exterior it has an extracellular component. This extracellular component interacts with the CAM or the cell adhesion molecule of another cell, and this is how the general adhesive contacts have the contact with other types of cells. Now the cell adhesion molecules, they can be calcium dependent or they can be calcium independent. The calcium dependent ones, they depend on the, so whenever the calcium ions are available, these molecules they have their function, but whenever the calcium ions are not available, these adhesion molecules they are useless. So there are two types of the calcium dependent molecules. One are the cadherins and other are the integrins. So now the cadherin molecules, they are between the epithelial cells and nervous tissues, whereas the integrin molecules, they connect the cell with the collagen, laminin and fibronectin. So you can say that the cadherin molecules, they attach one cell to another, whereas the integrin molecule, they attach cells with the extracellular matrix components. Now the calcium independent ones, they are the neuronal cell adhesion molecule or neural cell CAM. So these NCAMs, they attach the neuronal and the neuroepithelial cells, and their function does not depend on the calcium ions, because if their function started to depend on the calcium ions, then we will lose the integrity of the central nervous system. So now we have the specialized cell junctions. The specialized cell junctions, they are divided on the basis of their area and on the basis of their function. So the area that they cover, they can be zonula, they can be fascia, or they can be macula. Whereas on the basis of their function, they can be occluding ones, they can be anchoring ones, or they can be communicating ones. So now the zonula, they are girdle-like or they are belt-like, and they cover the entire perimeter of the cell. The entire perimeter of the cell is covered by zonula. Whereas the fascia, it is band-like and it has limited length. It only covers a small area, or you can say a strip-like junction. Whereas a macula, it is a spot-like and it is circular or oval in shape. Let me show you these three. So look at this particular diagram. This is a cell and this is a zonula cell junction which covers the entire perimeter of the cell. Whereas this one, this is the fascia junction which only covers a limited area. It is like a strip-like junction. Whereas the macula one, it is circular and small and it has the smallest area of coverage. So classification according to the function now, they can be occluding ones which actually occlude the area. They do not allow any paracellular communication, whereas the anchoring ones, they anchor or provide a support, and the communicating ones, they provide metabolic and chemical and electrical linking between two cells. So now the occluding junctions, they are actually formed when the plasma membrane of the adjacent cells, they come into contact with each other and the cells fuse together. As a result of which these junctions, they actually impede the barrier. They are virtually impermeable and they make a diffusion barrier. They block the paracellular routes, as a result of which water and other molecules cannot move through them. Also, they prevent the lateral migration of receptors, that are specialized cell membrane proteins. Now, these junctions, they maintain the polarity of the cells as well. They're especially found in the epithelial tissue, and their area that they cover is a zonula, that is, they cover the entire perimeter of the cell. So now look at this diagram to actually visualize another type of cell junction, which is the zonula adherens. So now the zonula, they actually cover the entire perimeter, but since I wanted to show you guys a little bit of the area, so I restricted it here. So now the zonula occludens, which are occluding type of cell junctions, on the cytoplasmic side, we have actin molecules, which are then attached to this portion, which is the zonula occludin 1, 2, or 3 proteins, which are further attached to claudin or occludin proteins. These claudin and occludin, they interact with the claudin and occludin of the other cell, and hence this is how the zonula occludins have their interaction with the other molecules. And since their name is zonula, so they cover the entire perimeter of the cell. So here we can see zonula occludens, which are belt-like junctions, and they limit the permeability of intercellular space, or they block the paracellular route, and they have focal fusion points between the adjacent cells. They are mainly present in proximal convoluted tubule of the kidneys, because we do not want any extravasation of the fluid in kidneys. They are present in the intestines and the urinary bladder. Since we do not want any water extravasation or urine extravasation or extravasation of the fluids, so we have the zonula occludens in these three specific areas. And since they have fusion sites, they have sealing capability. This is how they block the paracellular route. And since I told you about the actin filaments, the zonula occludin 1, 2, and 3 proteins, which are then attached to occludin and claudin proteins, and the occludin and claudin of the other cell, they interact with the occludin and claudin of another cell, and this is how they block the whole paracellular route and passageway. Now we have another type of cell junctions, which are the anchoring junctions. They're also called adhering junctions. They interact or they adhere the cell to cell or the cell to basal lamina. They can be zonula adherens, fascia adherens, macula adherens, and hemidesmosome. Macula adherens are also known as the desmosomes. Let us look at them. So now look at this diagram to understand the zonula adherence. So we have this cell, and as we know, zonula, they cover the entire perimeter, but for our understanding I have made it a little bit shorter. So on the cytoplasmic side, we have actin molecule. Attached to the actin, we have catenin and vinculin. Attached with these catenin and vinculin, we have e-cadherins, and as we know, cadherins are general cell adhesion molecule which depend upon the calcium ions. So the e-cadherins, which are attached to the calcium ions, now another e-cadherin which is further attached to catenin and vinculin on the cytoplasmic side, which are attached to the actin filaments. This is how a zonula adherens takes place. So it is a belt-like, it covers the entire perimeter of the cell and the intercellular gap between the cells. So this area, that is the gap between the two cells, this area is 20 nanometers. This area is 20 nanometers, and there is a paracellular route. So now we have catenin, and so now firstly we have the actin filaments, which are then attached to anchoring proteins catenin and vinculin, which are attached to e-cadherin molecules, and e-cadherin, they depend on the calcium ions, and this is how the zonula adherens work, and they are a part of another complex, which is the junctional complex. We'll study that later. And the most specific area of their occurrence is epithelial sheets. Now, fascia adherents, they are similar to the zonula adherents, and fascia means a band. It covers a limited area. It is a belt-like, it is a girdle. The fascia adherents, they are actually a band-like or a strip-like junction. That is, they have a limited length as compared to the zonula one, and they are especially abundant in the cells like the cardiac muscle cells, smooth muscle cells, and neuroglial cells, whereas the zonula ones, they were present in epithelial cells. Now we have the macula adherens or the desmosomes. They are very important but very easy. So now the intercellular gap at the level of the macula adherens is 30 nanometers, and we have an attachment plaque. Let me show you the diagram. So now look at this diagram. If you understand this, you will understand the area of a desmosome or a macula adherens very easily. So now we have this attachment plaque, which is attached on the interior or the cytoplasmic side with the intermediate filaments, which can be keratin filament or desmin filament. Now, the attachment plaque is made up of desmoplakin or placoglobins, and it is attached to these E-cadherin molecules. These E-cadherin molecules, they can be made up of desmocollins or desmoglins, and since they are cadherin, so they are calcium dependent, and the cadherin of one side interacts with the cadherin of the other cell, and this is how a desmosome is present, and it allows cell to cell junction because it has cadherin molecules attached to it. And the intercellular paracellular route at this area of desmosome is 30 nanometers, whereas in this area it is 20 nanometers. It is a little bit extended over here. So now we have desmosomes, which are spot-like or welt-like. They are circular in arrangement. They are especially abundant in cardiac muscle cells. The gap at their area is 30 nanometer, but the usual gap is 20 nanometers. They have an attachment plaque which is made up of desmoplakin and placoglobin, which is written over here. Oh my god, desmoplakin and placoglobins. And the intermediate filaments that are attached to them, they can be desmin type or they can be keratin filaments. And the shape that they make is a hairpin turn. Now, the cadherin molecules, they have desmocollin and desmoglins attached to them, and since they are cadherins, so their linkage depends upon the calcium ion. This is how easy was macula adherens, and they are mainly found in the cardiac muscle cells. Now we have hemidesmosome. So now we have a hemidesmosome, which actually means a half desmosome. On the cytoplasmic side, it is attached to the intermediate filament, which are inserted, which are make the hairpin-like junctions, and they are attached to the attachment plaque. This attachment plaque in the hemidesmosome, it is made up of desmoplakin and pectin, and these are attached to the integrins. Now the integrins allow cell to basal lamina connection. These integrin molecules, they are also calcium dependent general adhesion molecules. They attach the plaque with the laminin and collagen type 4 of the basement membrane, and this is how a hemidesmosome has its function and its structure. The basic function of hemidesmosome is to link the cell with the basal lamina via integrin proteins. The intermediate filaments, they can be keratin filament, etc. Now we have the communicating junctions. The communicating junctions, they attach one cell to another for the exchange of the material, and they provide tubular passageways. They are mainly there to allow cell to cell communication for the movement of ions and the small molecules between the two cells, so cells can be metabolically or electrically linked with each other. And these communicating junctions are also known as gap junctions. Now these gap junctions, they are also known as macula communicans. Communicans means allows communication. Macula is a spot-like, welt-like, and it is very circular type of junction. The intercellular gap at the area of gap junction is 3 nanometers, and they were also known as nexuses. The main area of presence of gap junction is epithelial cells, but they can be found in muscle cells, they can be in cardiac muscle cells, smooth muscle cells, neurons, astrocytes, osteocytes, which are bone cells, etc. But the one type of muscle cell which lacks the gap junction are the skeletal muscle cells. Now we have the gap junction. The gap junction is made up of 6 connexin molecules, 1, 2, 3, 4, 5, 6, and these six connexins, they combine together to form connexon. The connexon of one cell interacts with the connexon of another cell to provide a complete channel. One has a half channel, other has a half channel, and when both of these combine, they form a complete channel. Two hemichannels combine together to form a complete channel, and in this channel we will have a total of 12 connexons. A total of 2 connexons and 12 connexins, because each connexon is made up of 6 connexins. So they are writing over here that they have They are also known as the nexus. The gap is 3 nanometer. They provide transmembrane channels. Two hemichannel connect together, and each connexon is made up of 6 connexons. And when two connexons combine together, they form 12 connexons in total. And two hemichannels combine, and this is how macula communicans has its total anatomy. Now the lumen is 1.5 nanometers in diameter, and they allow the movement of ions, AMP, GMP, amino acids, and small molecules. The main function is to link one cell with another cell to provide electrical as well as metabolic communication. There are 20 different types of connexin proteins that can be present in the connexon molecules. And now a connexin, it opens or closes. It does not remain always open or always closed, but it depends on certain factors. The regulating factors, they are calcium ions and pH. Normally, when the calcium ions are low, connexins remain open, but when the calcium ions are higher, the connexin become closed. And calcium ion and pH have opposite relation. When calcium ion will be higher, pH will be lower. So main function is to provide electrical as well as metabolic coupling between cardiac and smooth muscle cells, but skeletal muscle cells, they have no gap junctions, and they provide a synchronous contraction of the muscle by electrical coupling. Nexus are especially numerous in embryonic cells for the exchange of information. As we know, an embryo has a large number of cells which can move, and these cells require a lot of signaling because they have to form organs and organ systems. So that is why nexuses are abundant in embryonic cells. Now, in the small intestine, we have another type of substance which is a junctional complex, which is made up of zonula occludens, zonula adherens, and desmosome. This is a very apical type of structure. Now we are done with the gap junctions, the adherent junctions, the zonula, etc. If you did not understand anything, please let me know in the comments. And if you understood anything, please let me know, and please like and subscribe to my channel. Thank you very much.